So what you see is just all the slides for mitosis, and I'm zooming in a little bit. I hope that's not making you seasick. And basically, it's the cell cycle in mitosis, and I just wanted to talk you through a couple of things. For sure, on the test, you're going to want to know this right here, that the three purposes for cell division, whether we're talking about mitosis or meiosis, are reproduction, growth and development, and tissue renewal, okay? So reproduction, that's meiosis, and growth and development and tissue renewal in eukaryotes, that is mitosis, okay? And the introductory slide, this one here, is showing you just the chromosomes and what it looks like when a cell goes through division. So down here on the bottom left is the nuclear envelope, and the chromatin has started to condense. And as you follow it kind of up this rainbow, you can see it getting more spaghetti noodle-like. And so that we're in prophase when this is happening. So this would be interphase at the very beginning. And then as the chromatin condenses into visible noodle-like chromosomes, this is all prophase. And so we're kind of going through early prophase to late prophase. And that is actually 6 of these images. And it isn't until this one right here where my mouse arrow is that we would call this metaphase. And that's where all the chromosomes that are made of paired sister chromatids are actually lined up at the middle. And this kind of shows you that chromosomes are shy. They don't line up perfectly for a picture every time. But if we were to draw an invisible line through here, we would call that the metaphase plate, the center where all the chromosomes are lined up. But because we're taking a picture and making something that's 3-dimensional, viewable as a 2D shape, we don't see it as a perfect line. And then as soon as you see those sister chromatids and the chromosomes start to separate, where it's almost like if you put your hands together and your palms together, and then you started to pull your hands apart but kept your fingers touching, and then pull them apart even further, and then your fingers separate, that would be the sister chromatids separating. And so this one, you know, all the way through, as long as it looks like fingers being pulled apart, that is anaphase, the separation of the sister chromatids. And then if you notice here, you can see how we don't really have, and maybe here, where the chromosome fingers are sort of starting to collapse and go missing. This is where actually we can tell that we are in telophase. And telophase is where the nuclear envelope starts to reform for each of the 2 new nuclei. And you can probably see on this next to last one that there's kind of an edge here, and that then there's definitely more of this like ovoid egg shape. And so that is the nuclear membrane that's reformed, you know, a nuclear envelope around the genetic material. And the chromosomes have really relaxed in here. And so then we would be back in interphase for these 2 cells. So what is mitosis? It is all of these stages here where the genetic material is condensing, moving to the middle of the cell, and then the sister chromatids, the paired copies of the chromosomes, are being split so that when 2 new nuclei form and when the cell membrane splits, and so we have 2 new cytoplasms, then we have 2 identical cells. And yeah, go for it.
Okay, on that slide that you showed us on reproduction, growth, and development and tissue renewal, what slide was that?
Slide 2.
Okay, thank you.
Okay, so here we are. Slide 3 just reminds you that most cell division is mitosis in eukaryotic cells. Most of your cells are somatic cells, non-reproductive cells. And so what do they produce? They produce clones of themselves. They go through life, a cell's life, performing cellular functions, and partway through that cell's life, it replicates its DNA in advance of the division that's going to occur later. And then it spends some more time taking care of cell work and monitoring the environment, and then it gets another signal, hey, it's time to divide, okay? And so then those steps that I was just describing, those are interphase, the cell doing cell work and when the DNA replicates. And then mitosis is the short period of time where the nucleus divides, or you can call that nuclear division. And mitosis is nuclear division that happens in somatic cells, non-reproductive cells. So it's happening in your liver, it's happening in your skin, it's happening in your heart, in your lungs, in your stomach, et cetera, et cetera. The only place where meiosis is happening, where we have DNA replication during interphase followed by a double nucleus splitting so that we end up with 4 cells instead of 2, that is happening in specialized reproductive cells that are producing gametes, the sperm or the egg cells, okay? So chapter 6 is about the cell cycle and somatic cell, non-reproductive cell division, mitosis. And chapter 7 is about specialized nuclear division that leads to daughter cells with half of the genetic material of the parent. Because what would happen if every egg or sperm had just as many chromosomes as the parent, and then the egg and the sperm got together? Then you'd have a zygote, a fertilized egg, with double chromosomes. So every generation, what would happen to the number of chromosomes? They would double, right? And that is, you've heard of trisomy 21 or Down syndrome? That's when just one chromosome that's relatively small doesn't split, and so there are 2 sister chromatids that go together, plus a 3rd chromosome that's normal, that isn't doubled up. And so one extra copy of that chromosome leads to the physical and genetic characteristics because of the extra proteins that are made in this, in every cell, because every cell has an extra copy of the 21st chromosome. So that's trisomy 21. A lot of the errors that happen when meiosis is occurring in chromosomes like 1 through 9, those are what we call lethal, in that it doesn't create a viable sperm or an egg. And so there's just, the egg isn't viable. And so it can't be fertilized, or if it is fertilized, the growth and development can't proceed from that point. So we want the right amount of DNA, and that means the cell has to monitor very closely what is happening to the chromosomes, whether it is mitosis for producing non-reproductive cells, or whether it is meiosis producing gametes.
How can you tell the difference between like a mutation that could potentially be like evolutionary or something like a positive thing versus that situation?
Well, most of your mutations are not heritable, can't be passed down from generation to generation. And the reason for that is they're happening in your somatic cells. So if your DNA mutates and it's in your liver cells or in your lung cells, the only person it's going to affect is you.
Okay.
And so if your cells mutate and the cell cycle, the cell correcting mechanisms don't catch it, then ultimately those mutations lead to cancer, which is simply proliferation of cells without all the correct cell cycle control mechanisms. And when we care about that cancer is when you get a tumor that affects the surrounding tissues because it's getting larger and dividing uncontrollably, and it's it's pressing on them, or when it metastasizes, and we would call it malignant because it can travel and the cancer can spread. And then how do you get rid of it, right? So when we target cancer with radiation and chemotherapy, we're targeting dividing cells. We're targeting the cell division process a lot of that time. And so that's why if we can target ways in which the cell gets ready to divide, and we can prevent cell division, then we might be able to prevent the growth of tumors, because tumors are cells that are dividing maybe more rapidly than they should, like the cell cycle is like going 10 times as fast, or without control mechanisms. And we cover, this slideshow covers that a little bit at the end, okay? So I'm gonna move ahead, and I don't wanna read everything for you, but I wanna show you this image. That's an actual microscope image of a replicated chromosome. So you see the two sister chromatids, and you can actually see the point where they're attached to each other. And you see how it's all like kind of popcorny? That's the millions of infoldings and spiraling of the lengths of chromatin into the condensed supercoiled chromosomes, so that they can be basically packaged for cell division. And so before replication, over here on the right, you see how it's showing a double helix DNA molecule. Each one of these sister chromatids is a double helix strand, okay? But then, if you remember from the beginning of chapter 9, they get opened up and copied, so that then we have two double helix strands that are identical, right? So they code for the exact same genes. If this is a gene coding for brown hair over here, then the other gene over here, that allele, the version of the gene, it's also going to code for brown hair because it's an exact copy. So the sister chromatids should be exactly identical because they came from the same DNA molecule that was split and copied. Okay, so I've already walked through this part, and I just want to remind you that cytokinesis is when the cytoplasm divides. And so the end of mitosis is not cytokinesis. The end of mitosis is telophase. And what happens simultaneous with that is the division of the cytoplasm, cytokinesis. You want another banana, baby? You already had one.
So when you were showing us the very first slide, so is the cytokinesis happening at the same time as the last picture where it says reformed nuclear?
Yeah, it is.
Okay.
So I'm gonna give her Peppa Pig. She's laughing. All right, so I'm gonna scroll ahead a little bit here and show you this, which hopefully from last week looks a little bit familiar, and it's just a graphic organizer to show you that interphase is all of this time, like 90% of this 360-degree circle, and interphase is where a cell spends most of its life. And the specific subphases of interphase are called G1, S, and G2. So G is gap 1, G1 is gap 1, G2 is gap 2, and S is in between those two growth and development phases when the DNA is replicated. So DNA gets replicated within interphase, and then as part of the cell's cycle, when it gets too big for its britches to operate efficiently, or when it receives signals from its neighbors, it goes ahead with the division process, and it splits its nuclei, which is mitosis, and splits its cytoplasm, which is cytokinesis. You may have seen in some of the videos a reference to prometaphase, and it used to be in textbooks that we identified 5 specific phases: prophase, prometaphase, metaphase, anaphase, and telophase. But now we just say, hey, prophase takes a long time, and you could see from that slide that I showed you that it takes a long time for the chromatin to condense into visible chromosomes. So we sort of have an early and a late prophase where prophase doesn't look the same throughout, and we can see how the chromatin is condensing. Okay, this video on mitosis, that's just a link to an old thing. This is from a textbook that I used to use at PCC where I pulled this in. That link isn't actually live anymore. Okay, so, but I did put a similar video. I loaded a couple of MPG files. It took a while, but I got them up, one for protein synthesis and one for meiosis, that are from this textbook file that I used to teach when I was at PCC. And that's where this image that I'm sharing is from as well. And it shows you a drawing down below of what it looks like, what the unique things are that are happening in mitosis. And then up on the top, you actually see images that were taken using a microscope with dye and special contrast to see the changes that are happening inside the nucleus. So this is zoomed in, you're not really seeing all of the cell, but you can see this blue blob on the left-hand side. That is the nucleus, and inside is the uncondensed chromatin. But we're late in interphase in G2, so the DNA has been replicated. And then you see this yellow stuff that's kind of moving around in like a web. Those are microtubules that are growing and expanding in order to get ready to dismantle the nuclear envelope and to provide the mitotic spindle, which is going to control the division of the chromosomes and separate the sister chromatids. So what are the things that you see happening in interphase? Well, there's a replicated centrosome. Normally, if you go all the way to the end, you see on the right, you see one centrosome set with a centriole pair. So in G1 and S phase, the cell only has one centrosome with this pair of microtubule centrioles inside. And that's where this microtubules grow from or originate, is the centriole pair. So in G2, we replicate that, the cell does. And we can see that the chromatin has been duplicated. So there's this huge mass of uncondensed DNA inside the nuclear envelope, and the nuclear envelope is intact. And the nucleolus is sometimes visible as a darker blob in images where you're looking at the cell, okay? Then how do we know that we've advanced from late interphase, where the cells are pretty big, into prophase? For one, you can see the mitotic spindle start to form if you have a really good slide, okay? So in this image, you can see how the yellow is starting to encroach around the blue nucleus. And can you also see that you kind of have these faint blue noodle shapes inside the nucleus? So that's happening in early prophase, where the chromatin starts to condense, where the mitotic spindle forms, where those centrosomes separate, and now we call that place where the microtubules are coming out of the centrosomes the aster. Just one more term that you wanted to know, right? So as we go into later prophase, but we're not to metaphase yet, the chromatin is fully condensed into chromosomes. They're completely visible as those noodles. And the two asters, where the microtubules are growing from the centrosomes, are really visible. And depending on the slide image, you can see the full mitotic spindle stretching across. And notice what's gone by late in prophase.
Nuclear envelope?
Yeah, the nuclear envelope is gone. So it starts to fragment in prophase and it disappears by late in prophase, okay? And that's because everything else, the other organelles are all getting squished to the side, and we've got to be able to spread those chromosomes in a human cell, all 46 of them, out and get ready to separate them, okay? So what we have happening is we have these non-kinetochore microtubules that are growing in length to expand the cell sort of north to south. You end up with pulls because it wants to kind of make an elongated cell so it can pull the chromosomes apart. And we have these kinetochore microtubules. They're cytoskeleton components that are actually bonded to the sister chromatids at the centromere, that part where the chromatids are attached. And their job is to either lengthen or shorten to push the chromosomes to the center. So how do we get from prophase to metaphase? The non-kinetochore microtubules lengthen the poles of the cell, and the kinetochore microtubules lengthen or shorten to push the chromosomes to the center. And notice they're all lined up. They're not in pairs, and there's not a particular order. It's just one by one, okay? So every single chromosome, even though there are 2 copies, because remember you get 1 copy from your dad of chromosome 1 and 1 copy of chromosome 1 from your mom. So you have 46 chromosomes, but you have a total of 23 pairs. So this sample here doesn't show you all 46, it just shows you 6. So this cell only has 3 chromosome sets, or a total of 6 chromosomes, compared to 23 sets or 46, but you're probably glad you don't have to count 46 across. Okay, so metaphase is just, hey, there's a pause where we check, where the cell checks to see, are all the chromosomes bonded to their kinetochores, and are they all lined up where they're supposed to be? What would be the point of splitting them apart if some of them weren't connected and in the right place? It would have been all this energy expended for what? Eventual apoptosis, programmed cell death. Sorry, you lose, right? So this is just where we're checking to make sure everything is lined up, okay? And that is actually basically the checkpoint in mitosis, is in metaphase, at the metaphase plate. So it's short, but there's a little pause here. Hey, is everybody where they're supposed to be? And then the sister chromatids separate from each other. They're no longer bonded at the centromeres. And anaphase is about sister chromatids separating in mitosis, okay? And they get pulled toward those asters at the poles. And so the hallmark of anaphase is just the fingers of those sister chromatids that are separating and getting farther and farther and farther apart. And how do I know when anaphase ends and telophase begins? It's really when I get this hallmark that the cytoplasm is also dividing at the same time. In an animal cell, it's the cleavage furrow. See how it's pinching in here? And we also will see a nucleolus forming. We'll see the mitotic spindle has disappeared, and the nuclear membrane starts to form. So this pinching in of the cleavage furrow tells you cytokinesis is happening simultaneously, which means you must be in telophase, not anaphase. The spindle is gone, the nucleolus is forming, and the nuclear membrane is forming. So those things that I just walked you through, those are the hallmarks of each of the steps of mitosis, okay? So if you have to like pause when I upload this on Zoom later, well, the YouTube version, and just go back through, just play it a time or two and kind of walk yourself through. Might help you with the lab too. Okay, and then if you want to play it back through, these next 2 slides, slide 12 and 13, are just zoomed in to show you what's happening, okay? That's all those are, just bigger. Okay, and this is a single image that shows you, hey, what do we mean by the metaphase plate? Do you see it's almost like a slide, a clear glass plate that's slid through here? If all chromosomes lined up perfectly, we'd be able to slide something through and have them all rest on it. But they don't line up quite that perfectly, but that's what the metaphase plate refers to, okay? And then there's just some labeling here that you might find helpful when you review. Okay, here's a good view of really zoomed in, seeing how the plasma membrane is pinching in to create the cleavage furrow so that the two cells can divide in an animal cell on the left-hand side. And then on the right-hand side, a plant cell, because it has a cell wall, doesn't actually do the cleavage furrow. The way that the cytoplasm gets separated is little vesicles of cellulose get created here to build the next cell wall, and we call that a cell plate until it actually divides the cell. And the cell wall and the cell can be added to make it a little bit bigger so that the individual cells can still grow throughout their lifetime. So just cool to look at. And this shows you a plant cell dividing. So you can see here we are in prophase, early prophase, where the nucleolus is still visible, but the chromatin is condensing. Now we're later in prophase and the nucleolus isn't quite gone, but the chromosomes are clearly visible. And now by the time we get into metaphase, there isn't an intermediate third image of prophase, but you can see that the nucleus is totally gone. And you can see those chromosomes are lined up. You don't get a good view of the asters like in the other image. But here you can see the chromosomes being pulled apart. Here's the mitotic spindle in the middle. And how do we know when we pass from anaphase to telophase? What do you see in a plant cell right through the middle here? You see that cell plate forming. And you also see that the chromosomes have started to do what? If you take your hands, remember, and you put them together, and then you start to pull them apart while your fingertips are touching, and then then pull your fingertips apart further so your hands are separated, and then collapse your fingers to your palms. That collapsing of your fingers to the palms is sort of what's happening in telophase, where the chromosomes are just relaxing onto each other, and then they're going to uncondense. Okay, I'm not focusing on this on the test too much, but prokaryotes don't do mitosis. They don't have linear chromosomes. They don't have to worry about, protecting everything. They don't have a nucleus, so they don't have a nuclear envelope to disintegrate, right? So they do have an origin of replication, if you remember talking about that last week. And so they do replicate their DNA, but then once that happens, the origins just get pulled by the cytoskeleton to the opposite ends of the cell. And then eventually the plasma membrane pinches off and we have cloned bacteria with cloned DNA. And that's bacterial replication in a nutshell. There are some bacteriologists who would get kind of upset at the simplistic view of it, but for what you need to know, it's just not linear and no mitotic spindle, nothing. There are some interesting things that are intermediates between what bacteria do and true, eukaryotic mitosis down here at the bottom in D, where we have like partial microtubules and partial spindles and some things that happen in protists and in fungi, which is part of the reason why those single-celled organisms are not grouped with bacteria. They're grouped with eukaryotes based on some of the ways that they behave, that they have organelles, that they divide, and so it's just interesting to consider that. But again, I'm not going to put that diagram on the test. Just know that there are some intermediates. Okay, so how do we regulate the cell cycle? How does the cell know when to replicate its DNA, when to go into mitosis? Okay, well, some interesting experiments were done where I have two summarized right here, where two cells, one that was in S phase, so like it was replicating its DNA, and the other one in G1, where it hadn't replicated its DNA. And as soon as in lab, the two cytoplasms were put together, the second cell started to replicate its DNA, which means there must be some molecule in the cytoplasm that accumulates and tells the cell, hey, time to replicate your DNA, because then you're going to have to divide. So this shows that there are, biomolecules, molecular markers in the cytoplasm that are going to tell a cell what to do next. And then experiment 2 says, hey, well, what happens if we take a cell that's in mitosis and we merge it with a cell in G1. So it hasn't even replicated its DNA. Look what that cell starts to do with only half the DNA. It enters mitosis anyway. So there's additional molecular markers in the cytoplasm that push the cell past S phase and past G2 into mitosis. I just thought you'd like that and think it was cool.
But what are they? Are they?
So there's one specific checkpoint molecule that we're going to get to in a couple of minutes.
Okay.
That accumulates. There's more going on than that. We could take 2 or 3 weeks to talk about just this stuff.
I was just curious if it was like, is it a protein? Is it an amino acid? Like, is it one of the biomolecules?
So in the one that we're going to talk about, it's a protein, and it's a protein like complex that
Good enough for me.
All right. So cells basically have a cell cycle control system or a cell clock that tell them what to do. And we say, hey, in this clock, you know, if this is a 24-hour clock, so to speak, where are the points where the cell says, hey, do we keep going or do we stop? Okay. And we call those cell cycle checkpoints. And just give me a second. All right, so moving forward rather than back. So do you see these 3 red gates here in this diagram? They're showing you where the 3 main checkpoints are. There's one in G1, so the first growth phase and interphase. There's one in G2, so for the cell to exit interphase. There's a G2 checkpoint, and then there's the M checkpoint, which I already told you about, which is metaphase of mitosis, where we're looking for whether those chromosomes are all lined up and attached to those protein kinetochores in order to divide. Okay, so for many cells, the G1 checkpoint seems to be the most important one, where basically if the cell makes it past the G1 checkpoint, it tends to breeze through G2 and M checkpoints and go ahead and fully divide. So if the cell doesn't receive the go-ahead signal, it exits the cell cycle, and that doesn't mean it dies. It means that it goes to a non-dividing state that we call G0, and G0 or G sub 0 for not dividing, okay? So this schematic just says, hey, if you pass the G1 checkpoint, then you get to go ahead and divide. And if you don't, then you go into this non-dividing state that we call G0. Some cells are pretty much in G0 permanently until they die. Why is it really bad to, you know, take drugs and kill brain cells? Because you don't get them back. They're in G0. It's you have nerve cells that you know, so my daughter who's 1, her brain cells aren't in G0. They're dividing and growing, right? But our brain cells, most of them are in a permanent G0 non-dividing state. So if you damage them, I don't mean non-functional, I mean non-dividing. Okay?
Non-dividing is not wrong.
It's non-dividing, and that's expected. So if you damage brain cells, it's really difficult to recover function if you have too much tissue death. So there's a difference between injured tissue and tissue death, and I'm not a neurologist, so don't press me too far on this, but we know that a lot of our neurons that send signals around our bodies are often in G0, and it can take a lot to stimulate nervous system tissue to regenerate neurons, which is why nerve damage takes longer to recover from than, you know, muscle or other soft tissue damage, or even bone damage. So other cells go into a G0 stage because there's simply not enough resources. If you're starving, you're not going to be dividing your cells as often because you don't have the nutrients that your cells need in order to divide. My son, who's 11, is definitely not starving because he just had dinner and he's in the kitchen again. All right, so here we get to what the question was about with what are some of the biomolecules that are regulating the cell. This is how you get through the G2 checkpoint, is with something called MPF, which stands for maturation promoting factor. And it's a 2-protein complex that basically triggers a cell's passage past the G2 checkpoint into M phase. So I'm gonna come back to that other graph here in a second. So here you see in the center that same cell cycle diagram. And can you see that CDK, or cyclin-dependent kinase, this purple rectangle with the dip in it, it's present all the time in the cell. Whether we're in interphase or mitosis, cyclin-dependent kinase is present. So cyclin-dependent kinase depends on cyclin binding to it. That's why it's cyclin-dependent, in order to make MPF, maturation promoting factor. And why it's called maturation promoting factor is as it builds up, it causes the cell to mature, to become ready, and to divide. And so when enough MPF is accumulated, there's enough of this biomolecule marker to signal the cell that it's time to enter mitosis, okay? So if you look up above, do you see how there's this, I don't even know what shades of purple to call this. This one's just a little bit more violet-y to me, but it has a point right here. So cyclin concentration peaks and plummets, and it stays at this low plateau, and then it peaks in mitosis, and then it plummets. Stays at the plateau, peaks, and then plummets. You get the pattern here? So as cyclin starts to achieve its peak, what is it that lags behind it a little bit and has a little bit of a softer peak? MPF. So what does that tell you is dependent on cyclin concentration increasing? MPF formation or concentration. So MPF is this combination of cyclin bonded with CDK, cyclin-dependent kinase. And when cyclin peaks, you see MPF activity peaks, and we're in mitosis. And then what happens right after cyclin concentration drops? MPF drops, and the cell is out of mitosis and in what? G1. And we can see this plateau of cyclin, and MPF is not even detectable. And then we get partway through S, and what do we see start to build up again? Cyclin. But we don't see MPF yet until all of a sudden, right here, at the end of G2 and going into mitosis, boom, we have a little bit of MPF, and then all of a sudden it's really detectable, and then it plummets again. So what's happening? Cyclin gets manufactured in the cell, starting in S phase. So there's some sort of molecular signal when the DNA it replicates that the cell then starts to manufacture more of the cyclin protein. And as the cyclin protein accumulates, it can bind with the cyclin-dependent kinase. It makes MPF, and as soon as we have enough MPF, boom, we pass the G2 checkpoint and the nucleus divides. Just go through that like 10 more times, right? And actually, if you've seen from last week, there's a set of only a few blank diagrams for exam 3. This is one of them. So you could practice labeling it and describing the process just so that it could make more sense to you. And hey, you won't have to do this, but when we do this class in person, part of the test is you have to know this called and label it and diagram it based on just like the 2 circles. So there's one blessing of like the online class. You don't have to do that, right? So there's some other things that stimulate the cell cycle. So the internal signal that I was talking about is M phase mitosis, the M checkpoint, has to do with whether the kinetochores and kinetochore microtubules are attached. And if they're not, there's a signal that says, metaphase needs to continue, we're not there yet. And so then anaphase won't start yet. There are other molecular signals that we would call growth factors, proteins that cells release that stimulate their neighbor cells to divide. So sort of like, we know that hormones are often sensed by individuals of the same species, while neighboring cells recognize proteins as growth factors too. And one of the examples in the text is platelet-derived growth factor, which is basically a growth factor produced by blood cells called platelets that says, hey, you should make fibroblasts. And fibroblasts are, their production is stimulated by the accumulation of platelets that gather around a cut. So when you cut yourself, what do you need to do to close over that wound? You need to clot first, but then you need to grow some new tissue. And platelet-derived growth factor produced by the platelets stimulates fibroblasts to grow a nice little patch over where you've injured yourself. So growth, repair, we hit both of those for mitosis, because how did my single-celled zygote become each of my kids? Through repeated cell division and differentiation. This is a schematic that shows an in vitro, like a cell sample in the lab, where it says, hey, put some cells on an agar Petri plate without platelet-derived growth factor, and what do? They just sit there. They live, but they just sit there. What happens when I add the growth factor? Oh, it signals them to grow and divide. And so it just says, hey, that's how we get these fibroblasts. So it's a particular type of growth factor in this type of cell. I bet you can imagine that there are all kinds of different growth factors that, function similarly for different cell types. Okay, so what are some of the things that control the cell cycle? I touch on them briefly here, and you've got in your learning assignment from last week, you learned some more about cell cycle control. So density-dependent inhibition is when cells are touching each other, they get a physical signal that says it's too crowded, don't divide, okay? Another signal that's external is called anchorage dependence, where a cell needs to be attached to something in order to divide. So what happens if either of these external signals that are basically density dependent are failing? So if the cell is crowded, but it doesn't get turned off by that crowdedness, then it can grow and grow and grow, divide out of control, and then what do you get? A tumor, okay? It's an overgrowth or proliferation of cells because they're not following many of the cell cycle controls, including density-dependent inhibition. And anchorage dependence, well, what happens if a set of cells breaks off from a particular tissue type and travels in the bloodstream or in the lymph system? Normally, it's supposed to just be taken care of, be digested by the body and be gone, right? Well, what happens if those cells that break off arrive somewhere else and start growing their kind of tissue type in a place where that tissue isn't supposed to be?
Cancer?
Yeah, that's another way that is malignant. That's when something metastasizes. Yeah, exactly.
So my mom's lung cancer spread to her spine.
And so it metastasized. Yeah.
So that finally made sense to me as to why it's still called lung cancer, even though it's in her spine. And
because of where it started out. Yeah. Yeah. My father-in-law's lung cancer did the same thing, traveled all over the place. So I know what you mean. So this shows you, hey, anchorage dependence, you got to be touching something in order to divide. Here's density dependent inhibition. So if we remove and there's a space, then the cells that are next to it will grow and divide and split in. That's normal. But on the right, you see cells that aren't anchored to a bottom layer, to a bottom spot. They're just growing and dividing. They don't care about contact. They would put 50 people in an elevator meant for 10. And it just doesn't work because there isn't enough space in an organism. My cousin, who's a few years younger than me, went through a second round of chemotherapy. She was a professor of Asian studies in Auckland, New Zealand, and had her first round of chemotherapy and surgery there. But when her cancer returned, last year, they decided to move back to the US because New Zealand doesn't often treat you as fully for round 2. And she went through surgery a few months ago, and she said they removed like a football-sized tumor from her abdominal cavity. And she said, and I won't tell you what other stuff they took out, but she appears to be in remission and doing well, and hopes to have many more years to take care of her kiddos. So yeah, I mean, we've got so much to treat cancer now. It's amazing. So what is it that makes something cancerous? Well, it's uncontrolled cell division. So cancer cells aren't responding to the body's control mechanism. They're ignoring those cell cycle checkpoints. They may not need a growth factor to grow and divide. They might just do it without. They might make their own growth factor, even though they shouldn't be. They may have a signal of the growth factor without the presence of the growth factor, sort of like cubic zirconium instead of a diamond, but somehow flashy, and it does the same job. They might have an abnormal cell cycle control system where something is always on or always off. There are genes that are called tumor suppressor genes that are supposed to limit cell division. There are proto-oncogenes that have other roles, and that's part of what you were looking at in the learning assignment for last week, and also what Lab 8 is going to be about. There's a section on that where there's a website you go to and you fill out a worksheet, and you do learn some great information there. So you're going to have fun with that. You will. It's good information, because for those of you going into the medical profession, cancer's not going anywhere. We're fighting it. We're looking for treatments. We're looking for cures. We're looking for things to help people enjoy their lives instead of being weak and exhausted and miserable from, you know, the standard treatments that we've been using for the last 2 decades, right? So just this slide just says, hey, let's look at some actual cells, some eukaryotic cells. And hey, here's a cell where you can sort of see the wiggly bits starting to form as opposed to speckles. So we would say that's early prophase. Here we see the actual noodles, late prophase right here. Down here, here's a cell where the chromosomes are lined up. So that's metaphase. Here they've started to separate, so it's anaphase. How do we know we've gone from anaphase to telophase? This is a plant cell, and do you see this yellow kind of blur down the middle? That's the cell plate that's starting to form. And can you also see that the chromosomes have kind of collapsed onto each other? That's the other clue that we're in telophase and not anaphase. So just something to help you. There's a part of the lab that I'm going to be posting tomorrow night that has more labels, more of these labeled, and some reminders of the hallmarks of mitosis, and then has you match them up. Okay, so that was, I'm trying to escape here. There we go. That was mitosis. And meiosis, I think what we'll do is we'll do some, we'll talk about meiosis for about 10 minutes. And then on Thursday, we'll come back to transcription and translation, because you'll have had a chance to look at the lab and work on it and watch some videos. And you'll hopefully have looked at some things on meiosis, and I can cover questions that you might have. Sound good? Just say yes. Okay, so notice this one isn't very long either, okay? So these, and a lot of this, see 21 through 25, they're just layers getting added. So really, really this is like 20 slides long, okay? That's all meiosis is 20 slides. And the other one that we just looked at, mitosis,
I think it was 36.
Yeah, it wasn't particularly long either. And we got through slide 23 of the DNA one, and that one, and it has some duplicate steps in it too. That one is really long, but there's a lot of vocabulary shoved in here and a lot of images to kind of show you the same thing, okay? So we'll come back and we'll do this one on Thursday, the second half, and there's a blank diagram in the blank diagrams file that corresponds with slides 49, 53, and 54. So it would be good to make sure that you've read through transcription and translation before we get to that PowerPoint, okay, for Thursday. If you've had a chance to look at it, that would be great. Okay, so meiosis is looking at special, oh, I forgot, I always forget that it does this. I made a flashy one once a couple of years ago, and even though I've updated the slides, I've never modified the way that it's flashy like this. So basically, meiosis is looking at reproductive cell division that is making gametes. So every single one of your cells has 46 chromosomes, 23 pairs of chromosomes. So that's your somatic cells, anything that's not a gamete, and even your reproductive cells that are going to produce the gametes. They're diploid or have 23 pairs or sets of chromosomes. And when we look at a karyotype, and you will do this a little bit for the second lab that you start way later this week, it's basically when we've condensed all of the replicated chromosomes, so all the DNA has been replicated, and so we see paired sister chromosomes. The karyotype is a display of the pairs of chromosomes from a cell. So you can, and they get lined up from the biggest to the smallest with the pair of sex chromosomes at the end, chromosome 23, and they're by size. So number 1 is the biggest and contains way more genes than, like, say, chromosome 22. All right, so what do we call a pair? So I'm not talking about the sister chromatids, which are replicated inside a single chromosome. But what do I call chromosome 1 from mom and chromosome 1 from dad? They code for the same genes. It's not that chromosome 1 from mom codes for hair color and how long your toes are, and chromosome 1 for dad codes for nose length and you know, how cute your ankles are, right? They code for the same proteins, the same genes. And so we call them homologous chromosomes or a homologous pair. So chromosomes in a homologous pair are the same length and carry genes controlling the same inherited characters. And so to make sure that we don't have too many genes that code for the same thing, what has to happen during mitosis? We have to split them so that the gametes have half of everything. You don't want 2 of chromosome 1, but none of chromosome 2. You want them split evenly, okay? So each pair of homologous chromosomes includes 1 chromosome from each parent, and the 46 chromosomes in a human somatic cell, again, like I've been saying, are 2 sets of 23. 1 set of 23 from the mother and 1 from the father. And a diploid is what we call all of those somatic cells. If you have 2 sets, you're 2N, and N is 23 chromosomes. So a shark and a turtle and anything that is diploid that reproduces with sexual reproduction and doesn't have an odd number of chromosomes, because strawberries are octoploid, they are 8N. They have 8 of every single one. Most organisms are diploid, they have 2 sets, okay? So for humans, the diploid number is 46. 2N equals 46, and N is 23. You have 23 pairs. Great white sharks have more chromosomes than humans, by the way. Okay, so in a cell in which DNA synthesis is occurred, each chromosome is replicated, okay? So we have paired sister chromatids within each chromosome, and then each of those paired sister chromatids is also part of a homologous pair. All right, so why do we use the 2N term to refer to diploid or diploid? Because there are 2 sets of every single chromosome, okay? And a cell undergoing mitotic cellular division, it never becomes haploid, because what happens when those chromosomes line up? What do? We separate the sister chromatids, but because chromosome 1 doesn't line up next to the other chromosome 1, they're in a long one behind the other line, every single cell gets a copy one copy of chromosome 1 that originated from mom a long time ago and one from dad. So when I made the eggs that got turned into my children, I have in every single one of my cells a copy of a chromosome from my mom and a chromosome 1 from my dad. And both of those replicated. And then they lined up and their sister chromatids separated so that in the daughter cells, I still have a chromosome 1 that came from my mom and a chromosome 1 that came from my dad. But in meiosis, when that happens, we actually have some interesting things that happen where we only have one copy, as opposed to mitosis where we have both copies, one from mom and one from dad. The way meiosis splits is we do split up that pair, and that's why meiosis has 2 rounds of division. We have to split up the pairs, and then we split the sister chromatids, where mitosis only splits the chromatids. And I am going to jump ahead for a second and show you this one right here, slide 13. This might help make more sense. In this scenario, because you don't want to look at 46 chromosomes, we're just doing 2n equals 6. Okay? I'm telling my son to go read a book, not watch TV. All right, so can you see that the original parent cell, if we call the long one chromosome 1 and the medium one chromosome 2 and the short one chromosome 3, can you see that that's how we have 3 sets? 1, 2, 3, okay? And basically, that's how we get 2n equals 6, because 2 times 3 is 6. Now we go through S phase, before meiosis and before mitosis, and so inside every cell, whether it's a somatic cell or a reproductive cell, what do I then have? I don't have more chromosomes. I have every single chromosome consists of the replicated genetic material, paired sister chromatids. So over here, I still have 1, 2, 3 sets of chromosomes. And notice in meiosis, I have the same thing, but they're joined together funny. I have 1, 2, and 3, but they're hanging out in these tetrads or sets of 4 chromatids together. That's something that's unique to meiosis, and we'll get to in a minute. So over here in mitosis, what happens to get to metaphase? What do those 6 chromosomes do? They line up individually at the metaphase plate, hallmark of mitosis. What do they do in the first round of meiosis? They're not lined up individually, are they? They're lined up in what are called tetrads. And so this is a hallmark of meiosis 1, and how, if you were clueless looking at a cell on a slide about where the cell got harvested from, that you would know that this cell was in metaphase 1 of meiosis. Okay, now we go through division here, where we have the cytoplasm divide and we have the sister chromatids divide. And what do we get at the end of mitosis? Do you see these daughter cells, 2n and 2n? What do they look like? Come back here to the parent cell. They look just like the parent cell. As long as there's been no mistake in the copying of the DNA in S phase, they are identical. They are clones. So the purpose of mitosis is to create identical copies so that that tissue type can be repaired or so it can grow, okay? What's the purpose of meiosis? Reproduction, making gametes, and we need to make haploid gametes, gametes with half the number of chromosomes. See, look down here at the bottom, how we've got only 1 copy of chromosome 1, 1 copy of 2, and 1 copy of 3. And notice they're not solid red or solid blue, and they're not all red or all blue either. There's a couple of things going on in meiosis that cause that. One of them is called crossing over, and one of them is independent assortment, okay? So there's 3 things that are really different about meiosis compared to mitosis. And one is these tetrads, these groupings of the homologous pairs that happen in meiosis 1 in prophase. The second is how those tetrads line up at the metaphase plate. And notice it's not all red on one side and all blue on the other. What chromosome 3 doesn't dictate the orientation of chromosome 1. And it didn't matter in mitosis over here on the left because there weren't homologous pairs joined together. They line up all separately. But here we have tetrads, and the way this tetrad lines up is independent from the way this tetrad lines up and independent from the way this tetrad lines up. So we call that independent assortment. And do you see how the chromosomes, the homologous pairs, how the sister chromatids that are next to each other have swapped? It's like they're changing arms from the elbow down to the hand, so to speak, and how the colors are kind of swapped over. Do you see that? We call that crossing over, and it's actually at a certain point on the length of a chromosome. Sometimes the DNA is actually swapped so that when we go through anaphase 1 of meiosis, the first round of division, do you see how we have chromosomes that are still the paired sister chromatids, but they're not all red or all blue? They don't have the exact same versions of the alleles that they started out with. And so we still have chromosome 1, chromosome 2, and chromosome 3. But do I have 2 copies of chromosome 1 anymore? Now your temptation is going to be to tell me yes, because you see the sister chromatids. But where's the other chromosome? Is it here? No, where is it? It is here. So what happened at anaphase 1 of meiosis? What happened to those homologous pairs? What did they do? They separated. So I'm going to back up here and I'm going to show you this cell. Now, do you see we've replicated the DNA and we have chromosome 1, chromosome 2, and chromosome 3? So we are 2n equals 6. Now, even if I was in G1, I would still be 2n equals 6, but what would I not have? I wouldn't have sister chromatids, but I'd still have these individual chromosomes. They would just be not replicated, okay? So I've doubled my DNA, and how am I gonna get to haploid? I have to split up my homologous pairs, and then I have to split up my chromatids. So that is why meiosis has 2 rounds of nuclear division, because I have to split things twice. I can't split my chromosomes and my chromatids in the same round of division. I have to do one or the other. And so what the cell does, all right, you see here, this is unreplicated and we're just looking at 1. So this is looking at chromosome 1. Now in S phase, the sister chromatids are replicated, but I'm still diploid. And then in meiosis 1, my homologous chromosomes separate. And as soon as they separate at the end of meiosis 1, because I only have 1 copy of each chromosome, yes, still with replicated chromatids, I am haploid. So you become haploid at the end of meiosis 1. And then meiosis 2 separates the sister chromatids. And so then what do I end up with at the end of meiosis is 4 daughter cells, okay? And it either has, for every chromosome, it either has the version that came from the parents, like from the grandfather, so to speak, or from the grandmother. And basically you're haploid because you only have 1 or the other. You don't have 2 copies of every chromosome anymore. So in a sperm or an egg, you don't have 1, you have 23 ones. You have 1 chromosome of chromosome 1, 2, 3, and so on, and either a single X or a single Y. And then at fertilization, what happens? All those copies join together so that then you have 2 copies of 1, 2, 3, etc. And then the 23rd set is either an X and an X get together from the egg and a sperm, or an X and a Y. And because that determines sex characteristics, we call that the sex chromosome, okay? So this slide walks you through what happens in the first round of division of meiosis, and this one walks you through the second. And I think that that is probably enough for today.
Yeah.
And that what I want you to do is watch some videos, read if you haven't read yet, and start working on the study guide, and we can come back and answer some specific questions about meiosis on Thursday, and then really hit transcription and translation.
Is the study guide due on